Can the optic nerve be protected? The new neuroprotection era in glaucoma research. Glaucoma has long been called the silent thief of sight, historically treated by focusing on intraocular pressure, the fluid pressure in the eye. But a growing body of research shows that glaucoma is not just a plumbing problem. It is also a neurodegenerative disease that gradually destroys the eye's nerve cells. Imagine your eye as a camera and the optic nerve as the cable that carries its images to your brain. In glaucoma, this cable gets frayed and rusty over time, not only from high pressure, but from an internal wear and tear process. In this article, we'll explain why that matters and how new treatments are trying to protect the neural wiring of the eye. We'll use simple metaphors, nothing too technical, so you can follow along easily. Retinal ganglion cells, the eye's messengers. Inside the eye's retina, special nerve cells called retinal ganglion cells, RGCs, work like telephone wires, carrying visual signals from the eye to the brain. Each eye has about 1.5 million of these cells, whose long fibers bundle together into the optic nerve. Think of RGCs like millions of tiny light bulbs along a cable. When light hits the retina, RGCs convert that information into electrical signals that zoom up the optic nerve to the brain. RGCs are crucial. Once they die, our vision is lost in those areas. They do not regenerate on their own. As one review bluntly puts it, glaucoma is marked by the irreversible loss of retinal ganglion cells, RGCs. In other words, if these cells burn out, the damage is permanent. A 2021 study of lab-transplanted RGCs emphasizes that because RGCs transmit visual information from the retina to the brain, their progressive loss results in fading vision and, ultimately, blindness. In everyday terms, losing RGCs is like cutting fibers in a cable. The signal can't get through, and you get a blind spot or fair-sized dark area in your vision. Because RGCs do so much work, they burn a lot of energy. They're packed with tiny power plants called mitochondria, and they need good blood flow and nutrients. This makes them shinny glass in a storm, delicate and easily damaged. In glaucoma, anything that weakens RGCs, from starvation of blood to chemical rust, can cause them to die. Glaucoma, more than just high eye pressure. Traditionally, doctors have measured eye pressure as the key glaucoma risk. High pressure can physically squeeze the optic nerve fibers as they exit the eye, like pressing on a cable at the wall. This pressure can block roads for nutrients, slow down the traffic of essential chemicals, and trigger cell damage. But scientists now understand that high pressure is only one piece of the puzzle. In many patients, something else is at work hurting those nerve cells, even when pressure is normal. Neurodegeneration and the brain. In fact, glaucoma is increasingly seen as similar to other nerve diseases like Alzheimer's or Parkinson's, but focused on the eye and its brain connection. Studies have found that damaging glaucoma can spread beyond the eye all the way into the brain's visual centers. For example, a recent review explains that people with glaucoma often show changes in their brain, such as thinning of visual cortex or altered neural connections, much like early Alzheimer's patients. This hints that glaucoma triggers a kind of domino effect of damage along the visual pathways, not unlike what happens with other neurodegenerative diseases. Mechanistically, researchers are finding shared culprits between glaucoma and brain diseases, things like broken mitochondria, chronic inflammation, and clogged nerve transport systems. In simple terms, if Alzheimer's is a problem of aging brain cells, glaucoma may be a related problem of aging eye cells, RGCs, and their brain links. Beyond pressure, inflammation, oxidative stress, and vascular factors. Because glaucoma is more than just too much fluid, other harmful processes are blamed when we see vision worsen despite good pressure control. One key factor is inflammation. The eye, like the brain, has immune support cells, glia, that can overreact when stressed. Stressed RGCs send out danger signals such as reactive oxygen species, free radicals, nitric oxide, and inflammatory proteins like TNF-α and interleukins. This can trigger chronic inflammation that ironically damages the very neurons it was meant to protect. Here's an analogy. Imagine RGCs as factories. When something goes wrong, like machinery overheating, the factory alarms, inflammatory signals, go off. If the alarm system is too sensitive or stuck on, it can end up hurting the factory itself, not helping it. In glaucoma, exhausted RGC mitochondria may flood the retina with reactive oxygen, oxidative stress that activates this alarm, causing friendly fire against nerves. One review on glaucoma neuroinflammation describes how broken mitochondria in RGCs can set off the immune system, leading to a sustained damaging response. In short, when RGC energy centers fail, they trigger a damaging inflammation loop within the eye. Vascular factors also play a role. The tiny blood vessels that feed the optic nerve can be sensitive. Eye drops that raise heart rate or conditions like diabetes and high blood pressure can affect blood flow to the eye. Low blood pressure, especially at night, or vascular spasms are linked to worse glaucoma because they temporarily starve RGCs of oxygen. For instance, one comprehensive review notes that reduced blood perfusion pressure and faulty blood vessel regulation likely help drive RGC damage. In our cable analogy, this is like having power fluctuations in the electrical grid. Even if the cable and camera are fine, if the power supply is shaky, the system falters. This is why glaucoma specialists often pay attention to cardiovascular health and sometimes even advise moderating certain blood pressure medications at night. Why pressure control isn't always enough. All these factors explain why some patients keep losing vision, even when their eye pressure is low or normal. For example, normal tension glaucoma is a common scenario where eye pressure never gets high, yet RGC damage and optic nerve cupping progress. Conversely, in some patients with high pressure, lowering it stops further damage, but in many others, damage creeps on. As one expert noted, despite apparently good pressure readings, disease can worsen in a number of patients. In other words, lowering pressure is necessary, but sometimes not sufficient. A meta-analysis of patient studies put it starkly. Doctors have observed that RGC loss often continues despite lowering IOP, meaning that treatments only focused on pressure may not be beneficial for some glaucoma patients. Think of blood pressure for analogy. Lowering blood pressure helps most high-risk people, but if someone is still leaking cholesterol plaques or has other heart risks, they may still have a heart problem despite normal pressure. Similarly, in glaucoma, we must also target the nerve itself, not just the fluid pressure. The search for neuroprotective treatments. Since RGCs are dying by many causes, scientists have searched for neuroprotective strategies, treatments that can keep these nerve cells alive longer or healthier. In simple terms, neuroprotection means anything aimed at preventing nerve damage or death. This new era of research looks beyond pressure. It asks, how can we shield the optic nerve from harm, regardless of the pressure? Researchers are exploring many avenues, from drugs to diet to bioengineering. Here are some current and emerging strategies being studied. Neuroprotective eye medications. Some existing glaucoma drugs might have nerve-saving effects. For example, brimonidine, an eye drop that lowers pressure, was hoped to strengthen RGC survival. Lab studies in animals showed promise, but human trials have so far been disappointing. An evidence review reports that to date, clinical trials of such neuroprotectors have failed to show clear benefits in people. Another drug, memantine, used in Alzheimer's, was tested in large glaucoma trials but did not prove effective. At present, manufacturers have not reported any significant vision benefit, so memantine is not part of glaucoma care. In short, while drugs like these are studied, none are yet a proven neuroprotective cure. Growth factors and gene therapy. Scientists have tried giving eyes extra growth factors, proteins that help nerves survive and grow. For example, nerve growth factor, NGF, or brain-derived neurotrophic factor, BDNF, can keep RGCs from dying in animals. Experiments involving viral gene therapy are in early stages. For instance, researchers can inject a harmless virus carrying genes for protective proteins into the eye. One phase 1 trial, GVB 2001, is even testing a gene treatment to relax eye muscles for pressure control, and similar approaches might deliver neuroprotective genes later on. These techniques are still experimental. The hope is to one day use gene vectors to make the eye produce its own protective agents, but it is decades from routine use. Stem cell and cell transplants. In theory, if we could replace lost RGCs, vision might be restored. Lab teams have made RGC-like cells from stem cells and injected them into animal eyes. In one notable mouse study, transplanted RGCs survived up to a year and even sent out axon branches along the optic nerve path. This suggests the retina can accept new nerve cells in animals. However, integrating them safely in humans is vastly more complicated. At best, cell therapy is an exciting research project. It is not a treatment available today, but it shows that researchers are even thinking about rewiring the eye in the long run. Nutritional supplements. Some nutrients are under investigation for nerve health. For example, citicoline, a brain chemical, has been tested in glaucoma. In a 2020 Italian trial, adding citicoline eye drops to normal therapy slowed visual loss in patients whose glaucoma was progressing despite good pressure control. Citicoline may help nerve membrane repair and mitochondrial function. Similarly, nicotinamide, vitamin B3, has shown promise. In a recent trial, high-dose nicotinamide significantly improved measures of RGC function in normal tension glaucoma patients. Early studies find a boost in nerve signaling after nicotinamide, though long-term visual benefits are still being tested. Other supplements like coenzyme Q10, ginkgo biloba, or antioxidants have been studied for glaucoma. For instance, systematic reviews on ginkgo found no clear evidence it improves vision or pressures. The bottom line, some vitamins may give a small functional lift to nerves, but none are proven cures. Always check with your doctor before adding supplements, since research is ongoing. Neuroenhancement. This means boosting the performance of surviving cells. An example under study is electrical stimulation of the eye. Small electrical pulses, like a gentle pacemaker, can promote neural activity. An upcoming clinical trial, called Viron, is testing transorbital electrical stimulation in glaucoma with optic nerve damage. The idea is that stimulating the retina nerve might wake up sleeping vision or slow decline. This is very experimental, but it shows the variety of ideas. From drugs to devices, researchers are trying to enhance what RGCs are left, not just keeping them alive. Lifestyle and systemic health. Though not a direct treatment, doctors note that whole-body health matters. Conditions like diabetes or poorly controlled blood pressure can worsen glaucoma, and conversely, healthy diets rich in antioxidants may support nerve health. For example, good control of blood sugar and blood lipids, regular exercise, which improves blood flow, and a diet rich in leafy greens and omega-3s are general measures that might help the optic nerve indirectly. There are no magic lifestyle fixes specifically proven for glaucoma, but cardiovascular health is certainly important. Smoking cessation, moderate caffeine, and good sleep to avoid nighttime blood pressure dips are usually advised. Neuroprotection versus neuroenhancement versus regeneration. It helps to define a few terms. Neuroprotection means shielding existing nerve cells from damage. It's like insulating a wire to prevent fraying or giving cells extra body armor. All the treatments above, neuroprotective drugs, supplements, growth factors, fall under this category. They aim to preserve the RGCs you still have. Neuroenhancement means boosting the function of nerves that are still alive. This could involve improving signal transmission or reviving weak cells. Electrical stimulation is an example. It does not create new cells, but tries to make the remaining nerve fibers work better, somewhat like upgrading a cable modem to send data faster along the same wires. Neuroregeneration means growing new nerve cells or fibers. This is the hardest and most futuristic goal, essentially rebuilding the optic nerve. It covers stem cell transplants and gene edits that encourage nerve regrowth. In our cable analogy, regeneration is like installing completely new wiring where the old cable was cut. Right now, true regeneration is not clinically available. We have no therapy that rebuilds the optic nerve in humans. All we have are protective and, to a small extent, enhancement strategies. But distinguishing these terms helps. Neuroprotection and neuroenhancement aim to help the nerves you've still got, whereas regeneration would bring back what's already lost. Why neuroprotection trials are difficult. Showing that a drug or supplement actually saves the optic nerve in glaucoma is surprisingly hard. Why? First, glaucoma progresses very slowly in most people. To prove a treatment slows nerve loss, you need large groups of patients, hundreds, followed for many years. Visual field tests, where you press a button when you see lights, must be done regularly, often every few months, for at least 4 to 5 years to detect small differences. Many studies simply haven't run long enough to show a clear benefit over standard care. Second, ethical and practical issues arise. You can't withhold pressure treatment to test a protector. Everyone is on good IOP control drops. So trial drugs are usually given in addition to pressure-lowering therapy. This means any vision loss is already very slow, making an extra benefit hard to spot. On top of that, about 30 to 40% of patients drop out of long trials because they move away, develop other illnesses, etc., which blurs the results. Third, measuring nerve health is tricky. We often rely on visual field tests or retinal scans, OCT thickness of nerve fiber layers, as indirect markers, but these can fluctuate and have measurement error. Detecting a small protective effect on nerve layers requires very precise instruments and analysis. As one systematic review noted, trials testing neuroprotective eye drops or pills in people have generally been inconclusive. In fact, a Cochrane review concluded flatly that, at present, there is not enough evidence to show whether any neuroprotective drug works in glaucoma. In drug development terms, work in this area is still at a relatively early stage compared to standard treatments. In summary, proving neuroprotection is slow, expensive, and uncertain. This is why, despite exciting lab results, the journey to clinical proof is taking time. What patients should and shouldn't assume. Given all this research hype, what should you as a patient take away? The good news is hope, but the reality is patience. Here are some practical points. Keep using your eye drops. Above all, don't stop your pressure-lowering medications or skip doctor appointments. Lowering intraocular pressure is still the only treatment proven to slow most glaucoma. Nothing in current research replaces the need for IOP control. Think of neuroprotective ideas as possible backup systems in development, not alternatives to your drops or surgeries. Be skeptical of quick cures. You may read about vitamins, herbal extracts, or devices online. Remember that supplements like ginkgo or large-dose antioxidants have not been definitively shown to save vision. These natural products are generally safe in moderation, but don't expect them to cure glaucoma. Always discuss any new supplement with your doctor because it may interact with medications or have side effects. Healthy lifestyle helps. Overall, while no diet will cure glaucoma, taking care of your whole health is wise. Control your blood pressure, eat a balanced diet rich in leafy greens and omega-3s, exercise, and sleep well. Good vascular health can only help your eyes, for example, avoiding very low blood pressure at night. These steps may not replace medicine, but they support neural health overall. No regeneration yet. You might hear headlines about optic nerve regeneration or stem cell cures. Be clear, these are experimental lab findings, not treatments. People should not expect new RGCs to spring back in the next few years. The media sometimes oversimplifies early research. For now, the phrase optical nerve cannot yet be regenerated is sadly true in humans. Stay informed and ask questions. Research is ongoing. If a new neuroprotective drug or therapy looks promising in studies, talk to your eye doctor. Clinical trials often recruit patients for eligibility. But until something is proven in large trials, news of a new therapy should not change your current care plan. Mental health. Coping with vision loss can be stressful. Sharing your concerns and being informed helps. Many patient groups recommend mindfulness or counseling to stay calm. Understanding glaucoma is partly a nerve disease can be frustrating, but doctors are increasingly focusing on nerve health too. Conclusion. Glaucoma research has indeed entered a neuroprotection era. Scientists now view glaucoma as a disease of the brain-eye connection and are exploring ways to shield retinal ganglion cells from harm. This has opened exciting avenues: drugs, vitamins, gene therapies, and even electrical devices aimed at protecting your optic nerve. However, it's important to remember that these ideas are largely at the experimental or trial stage. To date, no neuroprotective treatment has become a standard medical therapy because proving efficacy is hard. What can you do now? Keep using your prescribed glaucoma treatments faithfully, follow your eye doctor's advice, and maintain overall health. Use this time to be informed, ask questions, and perhaps participate in trials appropriate. In the meantime, the scientific community is working hard, combining insights from neurology, immunology, and cell biology to hopefully bring better therapies in the future. By understanding glaucoma as more than just pressure, we are one step closer to preserving vision. The road is long, but the new focus on neuron health gives patients reasons to be optimistic. even as we wait for solid treatments that truly protect the optic nerve. Sources: recent reviews and studies on glaucoma neuroprotection, systematic support the points above. These include open access articles on retinal ganglion cell loss, clinical trials of supplements, and systematic reviews of neuroprotective strategies. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.